everyone, and thank you for joining DNA LC Live. My name is Megan. I am an educator for the DNA Learning Center over in Cold Spring Harbor. Thank you so much for joining us. This will be premiering, so if you're joining us live for the premiere, feel free to leave any questions that you have in the chat box. I'll actually be moderating in real time, so I'll get back to you with those answers. And if you're joining us later on, feel free to leave any questions or comments you have down below in the comments section, and I can also get back to you then as well. All right, so what we are going to be talking about today is going to be Gregor Mendel, his work, and also I'm going to show you how to use a Punnett Square. All right, so let's get started. So Gregor Mendel was actually an Austrian monk in the 1800s, and what he did was, while at the monastery, he noticed that certain plants in the garden there had different traits, and specifically that plant he was talking about was this one right here, pea plants. So he noticed that there were lots of different types of traits that these pea plants could have. He noticed that some of them grew very tall, while others stayed short. He noticed that some of them had green peas, while other ones had yellow peas. He noticed that some of them had pea pods that were puffed, while others were smooth. So he was noticing all these differences, and he wanted to understand why those differences happen, and also how are they passed down to the plant's offspring. So he decided to take a closer look at these plants and start some experimenting. So let's take a closer look. So the flowers on a plant actually have both parts to make pollination or fertilization possible. Okay, they have both male and female parts. So it is possible for a flower to actually self-pollinate. However, there is something called cross-pollination. When we think of pollination, we usually think of things like bees and other insects who actually will come in and land on the, or I should say in the flower. They're trying to collect pollen as well. And as they leave the flower and land on a new flower, they are going to be dropping some of that pollen into the new flower, allowing for that cross to happen. Now, if you think about it for a moment, if we were to take, for example, pollen from a red flower and deposit it on a plant that has a white flower, what do you think the offspring would be? What color flowers do you think they would be? It may be a pretty common answer for people to assume it might be somewhere in the middle. It might be pink. But that's not what Gregor Mendel found when he started doing his research. He decided to take flowers from the pea plants and actually remove certain parts of the flower so that the flowers was not able to self-pollinate. He wanted to be able to have a controlled experiment where he was choosing which plants were pollinating with which plants. So he would actually collect pollen using a paintbrush and choose where he was depositing it, in what other plant. So let's take a look at what he found. So what he did, we call this the Pea generation, the parent generation. He took a purebred tall plant, meaning that it came from lots and lots of tall plants, and he bred that with a short plant. Okay. Now take a moment and think about it. What do you think the result would be for this cross? Do you think it might be 50-50? You'd wind up with half tall, half short? That's what I would think, right? But no, that's not what we found. We found that during this first generation, that 100% of the plants that were offspring from that cross were tall. It's like the trait of being short disappeared. So that was pretty interesting and very surprising. So he did another experiment, and he took plants from this generation and he bred them together. And what he noticed after that, well, what would we think? We think they're all going to be tall, right? Well, what he saw was that in that second generation, we had 75% that wound up being tall, 25% being short. So it's like the short trait actually skipped a generation and reappeared in the second generation. All right, that was a really big find, but he actually wasn't fully respected for his work back then. We didn't understand how important this was going to be in future research. He's actually now known as the father of genetics because of his work, but it wasn't noticed during his lifetime. What Gregor Mendel had actually helped discover was the fact that certain traits are considered dominant and other traits are considered recessive. So if we look at this chart here, we can see that one of those traits was dominant. Now, what comes to mind when you think of the word dominant? I tend to think of something that's strong, something that takes charge, and we can see that one of the traits showed up here a lot more than the other, and that was the tall trait. So these were our dominant traits, tall, which means that short was a recessive trait that actually at times it was able to hide until it showed up in another generation. All right, now we know that you receive half of your DNA from your biological mother and the other half from your biological father. You run the chance of inheriting either a dominant trait or recessive trait, but actually at this point, I'm going to stop using the word trait and I'm going to use the word allele. So you have a chance of inheriting a dominant allele or a recessive allele. Now, when trying to illustrate this, we use letters to represent the alleles. So for a dominant allele, we use a capital letter. And I'm going to use the letter T. Two reasons why I'm using the letter T. First of all, because we're still talking about being tall or short, and tall was dominant, so I'm going to use a T. The other reason why I use a T is because when we go to represent the recessive, I'm not going to use an S. For short, I'm going to use a lowercase t. T is a good letter to use because the capital and lowercase look very different, unlike other letters like S, where they look the same, or W. So that can get a little bit tricky when you're trying to do a Punnett Square, which we'll get to. So you run the chance of receiving a dominant allele or a recessive allele. So let's just use this example here. Say you do receive a dominant allele from dad and a recessive allele from mom. This right here is what we consider your genotype. It's a representation of those alleles, the genes. That's what helps me remember that. Okay, so that's the genotype. We also have to figure out the phenotype of this plant. Phenotype. Phenotype is the way that trait is going to express itself. What is that going to physically look like? And in the case of this plant, this one's a little tricky. This plant has a dominant allele and a recessive allele. So is this plant going to be tall or is it going to be short? Remember, which one is dominant? Which one is going to take control? Which one's going to take over? It's going to be the dominant one. And we know that tall is dominant. So this plant here is going to be considered tall, or is going to be tall, I should say. Okay? Now, when you have two, we're going to take it one step further. When you have two different letters, a capital and a lowercase, we call that heterozygous. That prefix hetero meaning different. Okay? This will come up a lot if you are taking living environment. They'll say, you know, a heterozygous genotype, and you're going to have to know what that means. So keep this in mind. Heterozygous means the letters or the alleles are different. Okay? Well, let's talk about the opposite now. I'm going to just take this. We're going to minimize this for a moment. Let's talk about some other genotypes and phenotypes we could see. So we already talked about capital T, lowercase t as a genotype. And let's talk about if we had two capitals, a capital T and a capital T. All right, so that's two dominant alleles. What would be, that's the genotype. What would be the phenotype? What would this plant physically look like? Would it be tall or would it be short? This one's a little bit easier because it has two dominant, so it's going to show the dominant trait, which we know is tall. All right, excellent. Let's do one more. We know there is the possibility then for the plant to receive a lowercase t. Oh, my green always gives me difficulties. A lowercase t and a lowercase t, okay? Again, that would be considered its genotype. The genes, the alleles. And what would this plant look like? It has two recessive alleles. Therefore, it's going to take the recessive trait. So this one is going to be short. And that's its, what do we call that? Phenotype. Physical trait. All right, excellent. Okay, so as long as we've got a hold on this, I just want to add one other thing. Remember how we talked about this being heterozygous, a capital and a lowercase. But what do we call it when we have what we just did here? When we have two lowercases or two uppercases? What would these be considered? They're not considered heterozygous. They have the same of each. So that is actually going to be called homozygous. All right. Okay. Now we are going to move on to Punnett squares and see what the probability is of two plants having a certain type of offspring. So let's take a look at that. So this here is a Punnett square, and we use it as a way not to figure out, a lot of people get confused. They think that if we do a Punnett square, it means that these are the four kids and that's the trait they're going to have. That's not the case. This is probability. We're talking about the percentage chance that the offspring will have a certain trait. So let's stick with the example of tall versus short. I'm going to give, we're going to say that the mother, or we're talking about plants, so we'll say one parent plant is homozygous dominant, and we're giving it two capital T's. So one parent is going to go on the top. The other parent is going to go on this side. Okay? Now let's say the other parent is, well, let's follow the example that Gregor Mendel did. Homozygous recessive. Okay, so two lowercase t's. What we do with the Punnett square is we go box by box. So we're going to start in this first box at the top. And what I'm going to do is I'm going to take one allele from this parent, one allele from this parent. So if I do that, I'm going to take a capital T from right above, and then I'm going to take a lowercase t from there. Moving on to the next one, I do the same thing. I go from right above, one parent, right to the side, one parent. And it's going to be the same thing, a capital T here, lowercase t here. All right, keep going. Now at the bottom one, capital, lowercase, and the last box, capital, lowercase. Oops, I want to do that in that color. Okay, so this is our probability of what a cross between these two plants will result in. So let's take a closer look at what that is. So I have 1, 2, 3, 4. 4 out of 4 come out as the result, capital T, lowercase t. That's its genotype. Okay, let's talk about its phenotype. What is this plant going to look like? We're talking about the height, tall versus short. What is this plant going to be? It has both alleles, but which one's dominant? Because it has a dominant one, it's tall. So this plant is going to be tall. That's its phenotype, its physical look. And I always like to add percentages when I'm working with this. So considering that all 4 boxes have a tall heterozygous plant, I'm just going to put 100%. Okay. Seems pretty easy, right? All right, let's try and do the next generation that Gregor Mendel worked with. So remember, he self-pollinated this generation. So let's take 2 of these, cross them together, and see what our probability is there. Let's see if our results are the same as what Gregor Mendel came up with. All right, so whoop. We have letters flying all over the place. All right, so remember, one parent is going to go at the top. The other parent is going to go on the side. And we're going to use the exact same cross that we just saw, the result. So we had a heterozygous parent there and a heterozygous parent there. Okay, so now let's cross these. Let's see what we come up with as our results. So we go to the first box. Okay, this is going to be different now. Come to the first box. I'm going to take 1 parent from the top, 1 parent from the side, and we see that we get capital, capital. Okay, next box. 1 from the top, 1 from the side. So we're seeing 1 lowercase, 1 uppercase. In these cases, always just make sure that you put the capital first. Okay? All right, down here, capital, lowercase. And lowercase, lowercase. Interesting. So we have a lot of different options, or possibilities, I should say, for this cross. Okay, so now let's go through the different genotypes that are possible here. I'm going to write, I like to be nice and organized, so we're going to write genotypes here. We're going to write phenotypes. And we'll include our percentages afterwards. So what are the possible genotypes? All right, so the first one we have is capital T, lowercase t. So I'm just going to write that here. Capital T is possible. Okay, remember that's a homozygous dominant. We have another option or chance here of capital T, lowercase t, which is heterozygous. And then our last one that pops up is lowercase t, lowercase t. All right, so we have 3 possibilities for this cross as far as genotypes are concerned. Let's fill in the possible phenotypes. Okay, physical look. Capital T, what is that plant going to look like? Is it going to be tall or short? It's dominant, so it's going to be tall. This one right here, what's its phenotype going to be, tall or short? It has that dominant trait, so it takes over. It is also going to be tall, so I'm not going to write it again. And then our last one, 2 lowercase alleles. Recessive means it's going to be short. So you have the possibility of a plant being tall and a plant being short. Let's do percentages quickly. Out of the 4 boxes, how many of them show a tall plant? 1, 2, 3, just 3. So 3 out of 4. What percentage is that? I always like to think of quarters. That helps me think of it. 3 out of 4 is going to be 75%. So there is a 75% possibility that the offspring is going to be tall. Let's check out, we only have one box showing the possibility of a short plant. 1 out of 4, 1/4 is 25%. Now, does that match what Gregor Mendel showed us before? 3 tall, 1 short? Yes, so our work with the Punnett Square matches his work as well. So you were able to illustrate this using examples of pea plants, specifically the trait of being tall versus short. You can use this as well for all the other traits he looked at, like the shape of the peas. He wanted to see what the traits were or how the traits are passed on, of whether the peas are smooth or if they're wrinkled. We can also do this with our own traits. Okay, there are certain traits within ourselves that are considered dominant and some that are considered recessive. So I hope you enjoyed taking a look at Gregor Mendel's work today. I hope you had fun learning how to use a Punnett Square, and make sure to check out Amanda's video as she uses Punnett Squares to create a creature. Thanks so much. Have a great day.